Hi, my name is Paul Fisher from the University of Florida IFAS Extension, and we're up here in one of the growth chambers at the University of Florida where we do different research projects. And the reason we're here is that in our course we're mostly focused on greenhouse production, but there's more and more interest in growing in controlled environments where we're basically looking at plant factories with LED lighting and all of the energy for growth coming from electricity. Now, what we're doing in this particular project, it just happens to be the experiment that's going on right now, is we're using our growth chamber kind of like a kitchen shelf, where we're looking at the performance of plants, the living salad, under different types of light, simulating what it would be like in a homeowner's environment. And so we've got two kinds of LEDs and two types of fluorescence here, and we're just going to be measuring growth and nutritional value and some other parameters about these plants. But it gives us an opportunity to talk a little bit about this kind of a growth environment. So, one of the things that we can look at, for example, is how much light energy we need to have an actively growing plant. And so these plants here are getting enough light to really be quite productive. They've been here in the growth chamber for about 2 weeks. But you can see these plants over here that aren't receiving as much light, and the difference in growth, where we've got a lot paler color, less of the red anthocyanin pigment, and if we measure the dry weight or biomass, which is a measure of total photosynthesis in these plants, we'll see that there's much lower dry weight in these plants receiving less light. You can also see they're kind of spindly and less leaf area and elongated. So we can measure the effects of light quantity, the amount of light. That's what when we've talked in the course about moles per square meter per day, that's light quantity in the photosynthetically active radiation range. We can also look at light quality effects. And so that's really, when we look at these 4 different types of LEDs or fluorescence, they differ both in terms of their efficiency of converting electrical light into photosynthetically active radiation, power light, being 400 and 700 nanometers, and also in their spectral output, the colors or the types of light, the wavelengths that each lamp type puts out. So we used a tool called a spectroradiometer, which measures the output of these lights at different wavelengths. And so here, for example, we have one of our fluorescent lights, and this is the wavelengths here, 200 through to 850 nanometers. Doesn't matter if you can't really read this, but the principles are what's important. And this black line is all of the outputs, the different peaks there from the fluorescent. And between 400 and 700 nanometers, that would be the output that would be for photosynthesis or plant growth. The shorter wavelengths are high in energy, but there are areas, for example, in UV. And in the longer wavelengths, that's really just far red light and heat. So it partly encourages elongation and also heating up of the plants. So you can see this is a fluorescent light output. And in comparison, this is the output from an LED, one of our LED lights. And you can see that most of the output is in this 400 to 700 nanometer range. So that means that it's very efficient at putting out the wavelengths that the plants would use for photosynthesis growth. And there's very little UV or far red light in here, indicating conversion into our power light range. If we look within the power light range, we've got our colors of the spectrum. And so these lines here represent blue, a peak for blue light around in the 400 nanometer range, and a peak for red around 650 or so nanometers. And you can see that the LED here has got a peak in the blue and quite a lot almost in the red range here. And the way that they'll actually produce a white LED like this is that they'll take a blue LED and they coat it to spread out the different wavelengths across the photosynthetically active radiation spectrum. So we've got different light qualities here, And those light qualities are going to have some impacts on plant growth. For example, if you've got a lamp that is very high in red light and low in far red light, you'll tend to get more compact growth, just as one example there. Okay, well, we can measure light in a growth chamber, and this is a light meter. This is quite a sophisticated tool here. It's a very good quality light meter, and the difference between a really good quality light meter for measuring plant growth versus a cheaper light meter is that it's going to be measuring the light in that photosynthetically active radiation range. So for example, if I put this over here on the side of the shelf, it's reading 13 micromoles per square meter per second. If I put it closer underneath, if I put it right underneath this light here, it's up to around 300 micromoles per square centimeter per square meter per second. So you can see there's a big distribution of light that very quickly decreases as we move away from the lamps. In comparison, this, for example, is a lux or foot candle meter, and this would not be useful for measuring light in photosynthetically active radiation for LEDs or fluorescents, because this is measuring the intensity for our eye, the human eye, and not for plant growth. And so there can be some big differences in the amount of PAR, photosynthetically active radiation light, for different amounts of lumens. So you can't really look at the lumen output from an electric lamp and know whether it's going to be a good light for growing plants. There are some other things that we can do when we're measuring the environment in a growth chamber. Obviously, temperature and relative humidity are very important. What we have here is we've got a small logger that is logging temperature every few minutes, and so we can see if the growth chamber is keeping the temperature stable over time. And this is also a useful tool. This is an infrared thermometer, and so we can look at whether these different lamps are causing the leaf temperature to be different between our treatments. And so, for example, if the lamp is putting out a lot of heat, we'd expect the leaves to be warmer. So, for example, here, if I take some measurements of some leaves under the LEDs, they're around 22.2 to 23.2 degrees Celsius. If I look down here at these leaves underneath the fluorescents, then they're closer to 24 or 25 degrees Celsius. And so what that's telling us is there's more heating of the leaves under the fluorescent lamps. So this is just one example. You can see that these are really like a retail potted plant, but we can also produce plants in trays, produce microgreens, leafy greens, and really it's a variation on this type of growing system when we have something along the lines of a plant factory or a vertical farm. And I'm sure as we start in the 21st century to get more into phytopharmaceuticals and really high plant quality, then we're going to see a lot more of this kind of production.